There are two types of gene regulation: positive gene regulation and negative gene regulation. Negative gene regulation occurs when the operons are usually transcribed and they're usually turned on and require repressor molecules to bind to the operator and shut down gene transcription. One such example is the TRP operon. The TRP operon is a repressible operon that is normally turned on and generally allows transcription to occur on its own. However, protein molecules called TRP repressors can bind to the operator region of the operon and shut down gene transcription. These TRP repressor proteins are created by the TRPR regulatory gene. When these TRP repressor proteins are bound to the operator, they are not permanently bound. It is because these TRP repressors contain allosteric sites that change shape depending on the concentration of tryptophan molecules. In other words, with higher concentration of tryptophan molecules, then more of these tryptophan molecules, as seen by these green circles, will interact with these repressors, and more of these repressors will bind to the operators and thus shut down gene transcription more. However, on the other hand, if you have a low amount of tryptophan molecules, then most of the repressors are inactive, and the gene can then be normally transcribed. The next operon that we'll discuss is the lac operon. It is an inducible operon. In the lac inducible operon, the repressor, coded by the LAC-I gene you see over here, binds to the operator and switches the lac operon off. However, the repressor contains an allosteric site, and when allolactose, an inducer molecule, binds to this site of the repressor, then the repressor's ability to bind to the operator is nullified, and it drops off. Now the gene can then be normally transcribed by RNA polymerase and create an mRNA. The next type of gene regulation we'll discuss is positive gene regulation. Positive gene regulation is when an activator is required to initiate transcription. One such example is also the lac operon. In the lac operon in E. coli, if glucose levels are low and lactose levels are high, then a molecule called CAMP will accumulate in E. coli, and these molecules, they bind to CAP, and this CAP molecule then subsequently binds to the promoter region with the CAP molecule to the promoter region of the lac operon. By binding to the promoter region, this increases the affinity of the promoter region for RNA polymerase, which directly increases the rate of transcription. Alright, so this is all prokaryotic gene regulation. Now we move on to eukaryotic gene regulation. In eukaryotic gene regulation, the first step is the unpackaging of chromatin. The chromatin is then subsequently modified by various procedures. One such procedure is histone acetylation or deacetylation. Histone acetylation occurs when the histones are combined with acetyl groups, and these acetylated histones then grip the neighboring nucleosomes less tightly, causing the transcription process to be easier to carry out. The acetylation and deacetylation directly regulates the control of transcription. Another process is DNA methylation. DNA methylation actually reduces gene expression and is responsible for epigenetic inheritance and genomic imprinting. So after the modifications to the chromatin are done, it then goes into transcription. Transcription itself is initiated when protein-mediated binding of the DNA brings bound activators in contact with mediator proteins that interact with proteins at the promoter to help position and assemble the transcription initiation complex at the promoter. RNA polymerase, when sensing this transcription initiation complex, RNA polymerase II can then subsequently transcribe the eukaryotic gene. transcription rate is directly influenced by the association with control elements and specific transcription factors such as enhancers. The next step is RNA processing. RNA processing RNA processing is carried out when snRNP molecules, which stand for small nuclear ribonucleoproteins, associate with other proteins to create a spliceosome. The spliceosome then excises regions of the RNA called introns, which are non-protein coding regions of the RNA. By excising these out and then joining the coding regions called exons together, this creates a continually coding mRNA molecule from the pre-existing pre-mRNA molecule. After an alternative RNA splicing, the mRNA can be transcribed into various other proteins or polypeptides, depending on which exons and which introns are treated as exons and vice versa, and which exons are treated as introns. After RNA processing, then the mRNA goes to the cytoplasm. After it goes into cytoplasm, the mRNA is then translated. Before translation, however, some of the mRNA is degraded. This degradation process is generally monitored by the UTR 3' region of the mRNA. After translation, then the subsequent polypeptide molecule undergoes protein processing. Protein processing has various steps depending on the protein, such as cleaving or the addition of phosphate groups. After protein processing, then it goes down. Then the subsequent protein that forms, some of these proteins are degraded after a certain amount of time, such as cyclins, because ubiquitin is attached to the protein itself, and then proteasomes enter and degrade the protein. And then the rest of the proteins then travel throughout the body to commit their various functions.